I want to begin by thanking Makasha and Nuguse for inviting me to present to you this morning, and I apologize for not being able to be there. The focus of my talk is going to be an international initiative known as the Chickpea Innovation Lab. A significant portion of our efforts are directed towards Ethiopia, but we also have activities in other countries. In total, we have activities in 12 different countries and 29 partner institutions, all focused on the issue of trying to address the constraints to chickpea productivity. We focus both on plant genetics and microbial activities. About a third of our funding derives from the U.S. Agency for International Development, but we have a wide range of other international partners, several different countries, private philanthropic organizations, non-government organizations, and industry partners. We all focus on chickpea, which is the second or third most important grain legume, depending on who you're talking to. In any case, it's vital to food security in much of the developing world, including several countries in East Africa, Ethiopia being the most important of them in a quantitative sense. Globally, legumes provide about 30% of the protein nitrogen in the human diet, and this clearly is an important rationale for legumes being important in agriculture. You'll see that nutritional content of seed is a reflection of their protein concentration, and that protein concentration is largely dependent on the legume's capacity for symbiotic nitrogen fixation. So you'll see themes of nitrogen fixation and protein nutrition throughout my presentation. In addition to those factors, we also focus on factors that limit chickpea production: abiotic stress, biotic stress, adaptation to soil stresses, and domestication, which might not seem like a constraint to chickpea production at first glance, but indeed, if you begin to sequence genomes of the wild progenitors of chickpea, Sisera reticulatum and Sisera kinospermum, and you compare that to genome sequences and diversity in the cultivated species, you'll see that about 95% of the genetic diversity and a very large fraction of phenotypic diversity was left behind in the wild systems when humans cultivated or began to domesticate chickpea about 10,000 years ago. And so the basis of our project is to go back into these wild systems, systematically collect the wild progenitors, develop pre-breeding populations, genetic bridges between wild and cultivated, and to begin phenotyping those populations and identifying genomic regions that control traits of potential agronomic utility. So our project begins in southeastern Turkey, where we spent about 10 person years collecting the wild material over about 100,000 square kilometers. What we're aiming for is to collect the seeds from the plants that exist in those locations and to collect the microbes that associate with those plants. The premise of the wild collections is that the natural environment will drive adaptation, that plants that come from regions that are drier or hotter or have more extreme soils will be adapted to those conditions, so that evolution will have done the job of selection for us, and our goal is to make systematic collections and find the phenotypes. Given that we're focused on the role of the natural environment in shaping the diversity of the wild species, we spent a fair amount of time characterizing natural environments. Every time we located a new site, and the image that's shown here is in southeastern Turkey near a city called Beriyestepe, we would sample and collect soil. So we collected a very large number of soil samples, ran ICP-MS, and so we know a lot about soil type variation at all of the locations that we sampled. Similarly, we collected information about the natural environment by placing small sensors in the soil that could measure temperature and humidity, and we collected the microbes in those regions. The first thing that we did as we collected material was to sequence the genomes using a RAD-GBS approach, and that allowed us to quickly distinguish the two wild species present in southeastern Turkey, Sisera reticulatum and Sisera kinospermum, and to divide those species into the genetic populations that they comprise. We spent a fair amount of time developing genomic resources because these will be important to take more sophisticated molecular-based approaches to breeding. We're developing 3 high-quality reference genomes based on PacBio and Hi-C sequencing. We have about 200 and 20 relatively deep Illumina genome sequences from these species, and about 2,000 rad GBS genome sequences. And this provides a resource to understand not only the genetic diversity of the wilds, but also, as you'll see, to allow us to identify genome regions that control traits of particular agronomic importance and to facilitate trait integration. To make a long story short, once we made the collections from the wilds of Mesopotamia, we had a long period of time making genetic crosses. Those genetic crosses resulted in populations, recombinant inbred lines in our case. We crossed into approximately 6 different cultivated recipients, and those were crossed by about 20 different wild donors. That material was moved at the F3 and F4 stage into various places around the world, and here we see those populations growing in Ethiopia, in India, at UC Davis, and in Russia, being screened for different traits. Now, these populations have an important attribute. They contain about 2 orders of magnitude more diversity, segregating now in semi-domesticated backgrounds, about 2 orders of magnitude more diversity than is found in the cultivated systems, and they are a remarkable resource for trait discovery. In addition to collecting those plants, as I mentioned, we've been focusing on the microbes. We collected the microbes both at the domestication center of origin of the wild species and the crops, but also globally around the world. And one focus of these microbes were the organisms that fix nitrogen with chickpea. Turns out a number of different species within the genus Mesorizobia. Initially, we collected several hundred genomes from southeastern Turkey, and we identified that there are 3 wild species of the microbe of Mesorizobia that associate with the 2 different wild species of Cicer, Cicer recurrenspermum, and Cicer reticulatum. So there's 3 symbionts at the center of origin. As we began to sample globally, we found a very different picture. There are about 28 distinct species of Mesorizobia that nodulate and fix nitrogen with chickpea globally, and one of the things we're interested in understanding are the consequences of this expanded diversity of the chickpea symbionts. We've also been collecting the microbiome. These are the organisms that are not fixing nitrogen, but they define microbial communities that associate with chickpea, often that are enriched from relatively low frequency in the soil to high frequencies on the roots, on the nodules, and inside the roots and inside the nodules of chickpea plants. And we've sequenced several thousand wild communities, identified several million wild taxa, and we've begun to assemble living culture collections of these taxa so we can use them as tools to try to look for traits like plant growth promotion or the ability to mitigate stresses for the plant. Now, in all of these cases, we're interested in traits. You can think about traits in terms of plant genetics or traits in terms of crop genetics. We'd like to understand the genes, or at least the genome regions that are linked to those traits, and then use that knowledge to improve crops in farmer fields. And so we've had a significant focus on phenotyping. Initially, we phenotyped agronomic traits for things like seed yield, plant biomass, shattering, harvest index, and so on. And we have identified a number of loci that control these agronomic traits in these wild by cultivated populations. I'm going to very quickly go through the different individual traits that we're working on beyond those agronomic traits. We have several different groups around the world focused on drought tolerance. This is the LisiScan platform at ICRISAT, but we also have activities in Turkey and at UC Davis and in Ethiopia, focusing from very applied aspects to very fundamental aspects to understand drought tolerance. We've identified relatively high levels of tolerance to reduced soil moisture and also changing vapor pressure deficit in the wild species, distinct from the traits in the cultivated species. We've had focus on heat tolerance. We've identified traits for heat tolerance, the ability to set seed under high heat conditions. In fact, the screening was initially done in the spring and early summer in Hyderabad, India at ICRISAT, where the temperatures are quite high. We've had a focus on ascochyta blight, major factor limiting chickpea production in many parts of the world, including in Ethiopia. We've started screening our wild hybrid populations, and we've identified a number of accessions that have levels of tolerance and some that potentially have high levels of resistance to ascochyta blight. This is a project that's in its early phases. We've also begun sequencing some genomes of the ascochyta blight pathogen in Ethiopia, and we're interested in doing a bit of population genetics to understand the specific targets for ascochyta blight resistance in Ethiopia. We have a significant focus on fusarium wilt. Two students, Danye and Sultan, shown here on the upper left, and a student from Pakistan, have focused on characterizing the diversity of the pathogen in Ethiopia. We've sequenced about 280 pathogen genomes. We know that there's a very high level of diversity of the fusarium wilt pathogen in Ethiopia. There are at least 5 distinct species. And we've begun screening both our introgression populations for tolerance and resistance to the ascochyta blight pathogen, and we've been using molecular genetics to map genes that control fusarium wilt resistance. We've also been working on pod borer, a very important insect pathogen in chickpea, and one for which there's essentially no effective resistance in the cultivated species. We've identified high levels of tolerance in the wild species, much higher than the best known genotypes in cultivated, and we've started a limited screening of our pre-breeding populations of these segregating wild by cultivated hybrids for tolerance to Helicoverpa, with some promising results to date. We've been focusing on nitrogen fixation. This is a particularly interesting story. We've determined briefly that wild species are highly responsive to their co-adapted or co-evolved symbiont, but they're very specific for that co-evolved symbiont. By contrast, cultivated species lack specificity. They'll associate with a wide range of different microbes, but they never perform particularly well. So wild species are much more effective at nitrogen fixation, but much more specific for the microbes. Cultivated species are much less efficient at nitrogen fixation, but much better at associating with a wide range of different microbes. And we're in the process of trying to identify the genetic control of these traits and develop near isogenic lines in cultivated species to test the hypothesis that wild alleles can indeed result in increased agricultural outcomes for nitrogen fixation. In parallel to work on the crop, we have a very extensive effort on collecting and analyzing Mesorizobia species as microbial inoculants. We have a large activity in Ethiopia with this objective in mind. I'll come back to this in a minute. And we also have a focus on developing genotypes that are tolerant to acidic soils, particularly acidic soils that have high levels of aluminum and low phosphate availability as a consequence of aluminum solubilization at low pH. And we have identified genotypes, land races in particular, in Ethiopia that have tolerance to acidic aluminum-containing soils, and we're in the process of mapping those traits for the purposes of introgression into improved cultivated genotypes. Sort of as a side project, but one that turned out to be very interesting, we started looking at total protein concentration in our hybrid populations, and we determined that we could find protein concentrations in the hybrid material that were up to twofold higher, up to 50% total protein than the best known cultivated genotypes. And this is a project that will be expanding significantly this fall by characterizing a large number of F5 lines to both map traits for protein content, understand the specific protein composition using mass spectrometry, and hopefully beginning to breed for increased protein content and quality. We've also had a focus on seed coat traits because seed coat traits, particularly condensed tannins and other metabolites in the seed coats, affect flavor, and we're interested in breeding chickpea for improved flavor attributes. And this is a an interesting and slightly complex area, but we've made good progress in identifying genes that regulate Seedcoat attributes. I should say that a fundamental philosophy in the project is to make all data available as soon as it's in a form where it can effectively be shared. We've deposited several terabytes of data to the National Center for Biotechnology Information, all under an umbrella bio project, and all of this can be accessed by anyone that's interested in that information. And we've shared this both indirectly by making it available, but also directly by working with other folks around the world. We've also developed a database in conjunction with folks at the James Hutton Institute in Scotland that will keep track of all of our germplasm and the basic genotypes in the material. And all of the material has been placed into the public domain in the International Gene Bank System, International Germplasm System. Now, I want to close by saying that the challenges that we're trying to address are too large for any one group or institution to solve. Even this project has insufficient resources to tackle these problems. It requires a huge breadth of expertise, which is one of the reasons we have so many partners. But it also requires country-specific focuses, because the challenges to chickpea production and the required genetics or the required microbes will vary by country, but also regionally within country. Now, one of the things that we've done to try to address the nature of the challenge is to develop partnerships. And I want to give you an example of this using Inoculum. We established a partnership with a multinational corporation in Argentina known as Rhizobacter Incorporation. Rhizobacter develops commercial scale liquid inocula for us that's quality controlled. And this information has, these inoculants have sufficient stability that we can ship them around the world and conduct controlled inoculum trials using the same inoculants in different parts of the world. And we'll be using Rhizobacter inoculants to test nitrogen fixation outcomes both in Ethiopia and in the U.S. And it's this kind of partnership that, in fact, has enabled us to do effective inoculum trials in different parts of the world here. I want to close by saying that although I've addressed a large number of different phenotypes, I'm beginning to think about these not as individual phenotypes but trait packages. For example, we're interested in nitrogen fixation, but we know that this has to involve both plant genetics and bacterial genetics. We're interested in seed protein concentration and quality, and we know that that the palatability of seed depends on flavor, so protein and flavor are a package, just like plant and bacterial traits for nitrogen fixation are a package. Moreover, nitrogen fixation is in fact the sort of molecular machinery to drive seed protein concentration. So at a broader level, seed protein content and quality is dependent on nitrogen fixation, and they're really not extricable from one another. They need to be thought of as part of a system of traits. Similarly, flavor depends on condensed tannins, so biochemistry and genetic pathways, and abiotic stress, drought, heat, cold, affect the capacity for nitrogen fixation in the plant and in the bacterium, and they affect seed development and therefore protein concentration. So all of these traits interrelate to one another, and our goal is to breed and develop genotypes with these interrelationships in mind. I want to close by saying that as the program moves ahead, it's clear that we need a corporate mindset. We still need to train and conduct academic research because it's part of our mandate, but we need to make sure that we keep our focus on the outcomes. We need to give priority to activities that are successful and dispense with those that are not yielding the kind of outcomes that we need, because our goal is to develop improved crops and microbes into farmer fields.